Oh, it says live. Yeah.
Yes.
Did it work?
Hi, everyone. We are now live. This has been a while.
Good gracious.
Yay.
Okay, so I'm going to update the WordPress site right now with the active link,
Okay.
just in case
All right.
anybody's watching.
Okay, I'll stop the broadcast for now.
No, go ahead. Go ahead. Just keep
Oh,
talking.
okay, I'll just start. Okay.
It's updated. We're good.
All right. Okay, hi, everyone. Sorry for the delay. This is our Google Plus Microbiology Journal Club, and we have finally overcome all of our technical difficulties and are live right now. And
Yep.
so you can see that Laura and I are very, very relieved.
We're very relieved.
It's been a while. Okay, today we are going to cover this really interesting paper called Obligate Bacterial Mutualists. Mutualism is, okay, I cannot read backwards, but obligate bacterial mutualists evolving from environmental bacteria in natural insect populations. And so this nice little paper also gave me a lot of material for silly little puns like bugs and buds, because it is about stink bugs. And so I am a big fan of puns and bad jokes. And if this starts to get really bad, I apologize.
That is okay.
Yeah, I'm such, like, I have bad humor, and that's the end of it. Okay, so let's just do a brief introduction. I'm Karen. I'm a PhD student at Simon Fraser University in Vancouver, Canada. And I study a bacteria called Francisella and its virulence factor, so the proteins and genes required to cause disease. And my interest in this particular paper is that I really love microbes. I enjoy all aspects of learning about microbes and talking about them. And so actually, yeah, so like just learning about these complex relationships that microbes develop is fascinating and fun for me. And so, okay, I will turn it over to Laura.
Hi, I'm Laura Williams. I'm an assistant professor at Providence College, and I think that my internet connection was the reason that we were having technical difficulties. So if I somehow fade in and out or something, I apologize in advance. And I am currently working on projects that have to do with bacterial lifestyle. So I'm really interested in symbiosis as a bacterial lifestyle. And apart from that, I did my postdoc at Duke University with Jennifer Wernegreen on mutualists of carpenter ants. So this is well within something that I've studied before and find really, really fascinating. So this was a great kind of, it was like a throwback for me. It hasn't been that long, but still, it was really neat to see what people are working on and asking this general question about where do these mutualists come from and how do they evolve, which is a big question in that field. So this is a neat paper for that.
So I'm sure that means that, Laura, you can give us some context of where the field has been at and what are the main questions. So are they still asking the similar questions, or how has that changed over time?
Oh yeah, I mean, so one of the big challenges with working on in the system of insect symbionts, which is a really, really strong field with a lot of excellent researchers who are doing great work. In that field, when that's your study system, one of the things that I personally found a bit challenging was that oftentimes when you have an obligate symbiont where their genome has reduced to the point that it's very small, which is not necessarily the case here, but that's often what we see with like Buchnera and the organism that I worked on called Blackmania. What you find is that it's really, you can't culture them. So as a microbiologist, that gets very frustrating because you have a lot of questions that you'd like to ask that require the organism to be able to be cultured in a flask. So you can change temperature or change media components and nutrients. But you can't do that with these because they're so dependent on their host insects that you can't pull them apart and work on them. Some people are working on this. I think there's some approaches to try to use tissue culture to do that, but it's not a widespread technique. So where the field really started to find a lot of inroads is with genomics. The idea that if you can't culture the organism, if you can't culture this bug that's in a bug, then if you can get at its genome, that's kind of the next best thing to being able to say, what are its metabolic capabilities? What is it doing? What can't it do anymore? Why is it dependent on the host? And of course, you can start to kind of reconstruct an evolutionary history once you have access to that DNA sequence information. And I think that means that currently this is still the big question, which is, can we trace back where these symbionts originated? What were the steps that led to them kind of being irreversibly a symbiont? Because certainly the organism that I study on, that's it. You know, when I studied Leishmania, it was like, it is not going to become an environmental organism anymore. It is committed to this symbiotic lifestyle. So the question is, where are the junctures at which that happened? And what are the steps that kind of commit a bacterial lineage to being a symbiont like this? So this is neat to kind of see somebody do this with a different type of study organism.
So in that sense, we're comparing the state that they are at now with their closest relative that they were at in terms of genetically that you can find, and then comparing and then using that as a reference for the number of steps that happened. Is that correct?
Yeah, I think that's a really accurate way to put it. So you can find their closest relatives who are free living or at a or are facultative symbionts instead of obligate symbionts. So that's a distinction that gets made. You can find some symbionts that are obligate symbionts and some symbionts that are facultative. And you can find symbionts that are not as reduced, so their genomes haven't gone through the streamlining process that you see with things like Buchnera. And if you can do that, then you can start to potentially try to reconstruct the steps of, well, when did they lose this particular amino acid synthesis pathway? Because then that means that now they're dependent on to get that from the host. And so that's what you're trying to do with genomics.
Well, that's a great segue into what we talk about in the actual paper. And so let's just jump in since we have we're kind of short on time now. So in this paper, as I mentioned before, talks about stink bugs. And so these stink bugs are green. This is not the best black and white image that I have here.
Ooh, wait, I don't know if you can see that. I managed to print it in color. This is the poor man's screenshot. There you go, guys. Stink bugs, if you've not seen them.
So those stink bugs are kind of, they're pretty common, and what the researchers did is they collected a bunch of stink bugs all around of this particular species. They called Platia stalli, and they collected them all around Japan, and they genotyped both the stink bugs and the symbionts inside the stink bugs and created genetic trees of the symbionts within the stink bugs and also the symbionts themselves. And so what they found is, okay, sorry, I'm getting ahead of myself. These stink bugs are gut symbionts, so they're located in these gut sacs, which is what you see here, inside the stink bug. And then the females, what they do is they deposit these smears or this smear on top of the eggs when they lay the egg, and then the little larvae kind of look on top for that little smear of excrement, and that's how there's a vertical transfer of the symbionts to the child.
Yeah, and I think that's an interesting feature of this system that's a little different from what the one that I studied was like, because you get a lot of times with these obligate symbionts, they are vertically inherited, vertically transmitted. With this one, when I was reading this, I was like, oh, okay, this is going to be a bit different from what I'm familiar with, because in the carpenter ants that I studied, you actually see, you not only have Leishmania, their symbiont, in their midgut, but you also can see them in the oocytes. Okay. So they in fact get passed they get passed from the queen directly, like the eggs have them. So they go through development with these Leishmania. But I do know that there are other systems like this one with the stink bugs and their symbionts, where the offspring actually acquire the symbiont, like you said, from kind of picking it up from the deposits on the egg sacs. The neat thing about that is that, as we'll get to, helps you actually do experiments with trying to figure out, which symbiont can they work with. But that's getting ahead of things. So that was a neat feature that I was like, oh, okay, this is different from what I know.
So while we're on the topic of vertical evolution, is it true that for vertical evolution, it's more favorable for symbiosis and mutualism as compared to horizontal?
So you mean the acquisition of a symbiont?
Yes. So for for a symbiont to, if they initially start in a different relationship, even antagonistic, for it to become mutualistic, is it more favorable in a vertical transmission context compared to a horizontal transmission context?
Yeah, I think I think broadly that's a very reasonable hypothesis to think about the different ways that transmission of symbionts could occur, or acquisition of symbionts, depending on which partner's perspective you're thinking about. So for instance, Wolbachia can be horizontally transmitted. And Wolbachia shows up, it's a reproductive manipulator. So it's in a way is you could consider it a reproductive parasite, because it's going to influence the populations of its hosts to its own benefit and not necessarily to that of the host's benefit. There's some arguments about possible benefits of being infected with Wolbachia, but I mean, broadly, I think that when you get something that's going to be faithfully vertically inherited, maybe there is something there that fosters a move towards a more mutualistic partnership rather than a parasitic one. I could see that.
Well, and in this sense, because it is a gut bacteria, there's no selection pressure in terms of reproduction. In that sense, it's directly transferred from the mom to the egg. So any benefit within their, I don't know, I would hypothesize probably favors a mutualistic relationship as opposed to anything else.
Yeah, and I mean, when people started studying this, I think one of the clues that they had that these were, nutritional mutualists or mutualists in general, was the fact that they were prominently located in the gut. So when you see that by under the microscope, you're like, there's got to be, there must be, I mean, it's getting nutrients, the symbiont is, but then you can kind of start to see where that relationship might build from. So, yeah.
And we also see that in the later experiments as well. So we'll come back to that. So as I mentioned before, what they did is they sequenced all of the variants in all the bug variants, and then all the different symbiont variants within the Japanese stink bug population that they could collect. And so what they found in my unfortunate black and white figure here is that all the ones on the Japanese mainland have a specific type, and so they call that symbiont A. And then in the different collection of the islands down here, they have different ones where they have B, prominence of B, prominence C, and then various ones that have C, D, E, F. And these are just different symbiont types that they collected from these stink bugs. And so what was unique is that when they tried to cultivate these A to F, they found that A was not cultivable at all on standard media, so just LV. And was it B?
B you could call. Yeah, B so, and I'll just do the quick, since I got it printed. So that's kind of what it looks like. And I just thought it was neat that, like you said, before we talk about, this is really interesting to me, that on the mainland, you're seeing just absolute dominance of this symbiont A. So one lineage is being found all across the mainland, just absolutely. I don't think they saw any variation in that. And then when you get into the islands, like the southwestern islands they're talking about here, what they're saying is, you've got B is prominent, but then they're seeing some other symbionts kind of creeping in there. And this one was kind of neat, this little outlier where there's one island where C is the real dominant one, and then you get a little bit of the others too. So that's kind of cool, because they're finding these different lineages that all look relatively related, because they all kind of cluster tightly together on a phylogenetic tree. It's not like one of them's way over here with E. coli and one of them's like way over here with something else. They're all kind of together related to these soil organisms, Pantoja. But it's neat that they found such a strong geographic trend in seeing like the mainland is just utterly dominated by the A lineage, and then when you get into the southwestern islands, you've got B lineage, and then some smattering of others. I thought that was neat.
Is this a really unique data set in the context of in sequencing mutualists?
Gosh, you know, within a single species, I think so. So within a single species of stink bug, the fact that they're finding some variation in lineages, I think is interesting. What I'm used to seeing from what I'm familiar with is kind of cospeciation on a really extreme level, where you get, you know, one species of carpenter ant has a particular species, if you will, or lineage of the symbiont, and they kind of, if you put their phylogenetic trees next to each other, they track each other really well. And so geographic, you know, you get ants with geographic range, but their symbiont lineages will always kind of cluster with the ant. So I don't, in my experience, with the system I worked with, I didn't run across any geographic trends like this. So that's kind of neat. And it probably reflects the fact that this is like a more nascent symbiotic relationship. So these symbionts are not as, their genomes aren't as reduced as the ones that I'm used to thinking about. And I don't think they gave any dates. I don't think they ever tried to do any dating of the lineages to try to figure out when the first infection might be happening. In part because I think there's still some fluidity to how these lineages might get infected. But anyway, so.
So they did, in relation to what you just said, they did say that the genotypes in terms of the bug were not indicative of which particular symbiont they were going to carry. So those weren't related. And I think that also speaks to what you were mentioning, that this relationship is still very nascent in nature.
Yeah, and I made a note about that. I just didn't have a chance to look at the supplementary figure. But yeah, you're absolutely right. They made a point to say when they looked at the mitochondrial genotypes, and their last sentence from that section is, this suggests promiscuous host symbiont associations, and especially in the southwestern island populations. So it's fortunate that they must have known to expect this to some degree, but it's kind of fortunate that they didn't just sample from the mainland, because then they would have been like, oh, that's boring. But then they got into the southwestern islands. It was really neat.
So in terms of, I think one of the things that I had in terms of cultival, so they only said that they grew it on LB, and they said standard bacterial media. Is that, would you really call that, at least for me, I found that I don't know if I could really call that uncultivable if they only tried one particular media.
Yeah, so the fact that that's exactly what I wrote to you. So A and B, they tried to grow it on LB at like 25 degrees Celsius, and it didn't, no colonies came up after a few days, and they were like, uncultivable. And, you know, okay, but we don't know that for sure. You could try some other growth media. You could try some other temperatures, you know, and it may be that these, one of the things that they note is so they kind of end up separating the dominant lineages A and B away from these other lineages C, D, E, and F that keep kind of showing up, but they're not dominating any particular population geographically. And A and B can't be grown on LB at 25 degrees Celsius, but they could grow the others on LB at 25 degrees Celsius. The other thing they found was that the genomes of A and B were somewhat smaller than the genomes of C, D, E, and F, but the sizes are still quite a bit bigger than what we're used to seeing with things like Bucknera. So it could be that if you looked at the genome, you'd be like, oh, if we give them this one thing, they'll grow then, you know, like, or something it might be a little easier to supplement them than it would be some that have gone to the extreme end of being streamlined. So yeah, I agree with you. I kind of made a little note on that too, that I was like, well, I don't know if you can completely say they're uncultivable just yet, but it's
Well,
certainly a difference.
yes, certainly a difference. And I think that's also difficulty in terms of vocabulary, because sometimes you want to make a comment and you're like, well, it's not entirely this, but it would make more of a point if I said this compared to if I said that. But that's not necessarily untrue.
Yes, yes, yes. It gets into the joys of semantics, which doesn't everyone just love that?
Yes.
But.
And so into their next experiment, which is actually really exciting. So what they did is they took the stink bugs and they sterilized the eggs. So they just sterilized that little smear excrement on top of the eggs, so the larvae no longer get there's no vertical transmission of the symbionts from the mom to the larvae. And so once they did that, they found that the bacteria, most of them died.
Mm-hmm.
So, sorry, not the bacteria, the larvae.
Mm-hmm.
So here they're examining adult emergence, and that just means like how many adults were born compared to control. So the control is here, and then I don't know if you can see that. Control is there, and then in different clades, the different clades are just, if I understood correctly, the different types of, were they bacteria? Or were they bugs?
Those were the hosts. Those are the based on their mitochondrial genotyping. Those were 5 clades that they identified in the hosts.
Oh, those hosts. Okay, so
yeah.
5 different types of hosts, and then in every 5, once they sterilized it, the larvae no longer were able to grow into mature adults, or no longer able to grow into adults. And so they made the point that these symbionts are necessary for adult emergence. And so after that, what they did is they started to do replacement experiments where they would take where they would sterilize the eggs and then add back one of the strains that they found. So they only did this with B, C, D, and F, presumably probably because those.
They did a separate experiment with lineage A. So they actually, I wondered that too, but they did in fact do lineage A.
Okay, they did do lineage A? Okay.
Yeah, they noted, they didn't show the figure for it, that's in the supplemental, but they had one line in the text where they said, we also did this with lineage A, and we saw the same thing. Because I was wondering about that too. So I was looking specifically for that. I'm like, did you guys not do it with A? But they did.
Okay, that's good. All right, I'm glad you caught that. And so what they did, after they did this, when they replaced each of the symbionts into this sterilized stink bugs, they didn't find any differences in the adults. So essentially, each of these symbionts could do the job of each one, like anything from B to F could do the same job that they were supposed to do, because in all of the features, if you're going back to the metabolic pathway that we were talking about earlier, they could do the metabolism for the bacteria or for the host that was needed. Mm-hmm. And so if you look at all of these graphs, none of them are significant, simply because they, once after the replacement, the bugs went back to being, with at least these parameters, back to being normal.
Yeah. I mean, that was really striking, the fact that you're seeing no appreciable differences in some of these fitness phenotypes that we're used to measuring to try to see, how is the host doing, either in the absence of a symbiont or with a different symbiont. In the absence of a symbiont, I mean, obviously they don't do well. The percentage they just don't have any adults emerging at all. But then, like you said, when they do this replacement, all these fitness phenotypes, the nymphal period, adult emergence, number of eggs laid by females, they measured the thorax width of the males and the females, and they're not seeing any statistically significant differences, regardless of whether or not they've got symbiont lineage B, C, D, E, or F in there, which is really interesting. So that means that, and that kind of gets back to what they were saying, that they think there's still some kind of promiscuous host symbiont associations going on. So it's not come to the point where we've gotten to like a bottleneck where it's like this host species has been co-evolving with this bacterial symbiont species for so long that now they've just got to have each other and you can't make any swaps in there.
And so what also was interesting is that when they tried this with different was it Pantoja?
I say Pantoja, and
Oh
funnily
yes.
enough, there was actually a debate on Twitter recently about this, and so there were a wide variety of responses, like I'm not even gonna try, but like, you know, it just always makes me think of the shampoo, I have to be honest, so the Pantene, but I say a Pantoja.
Pantoja, okay.
Yeah.
Different species of Pantoja. They still saw, like one of them was not significant. The other one was strange. It was significant, but they still had a large number of adults emerge. So it wasn't that they had less, but it wasn't that they
Right.
didn't have any at all.
Well, the interesting difference in those is that the two Pantoja species they tested, so when they took the hosts that usually have symbiont lineage B, and then they replaced it with either a Pantoja species that's typically found in soil, or a Pantoja species that's a human pathogen. And I think that's correct. It was a human pathogen. And they're finding a difference, or at least okay, it may not be a pathogen, but it was isolated from human wounds.
Oh,
So but the soil one, the one that was isolated from soil, did better. So the one that where you still saw adult emergence rates, you still saw adults emerging, but the rate was significantly lower than the other, was the one that was isolated from human wounds. So I don't know if it's possible that, you know, that there's a lot of variation within a genus. So it may just be that particular species of Pantoja is just not quite as adapted to this symbiotic lifestyle with an insect. So, but that was, I mean, the fact that you can replace the symbiont lineage with a soil microorganism from the same related clade and have it be no different than if you had the original symbiont lineage, that's pretty cool. So.
no, that is pretty cool. Well, and the soil microbes would have been, I guess they would have been there. So then it's the best part about this paper is the fact that you can take and there's this free relationship where they're in the soil, but then they can also be in the bug.
Mm-hmm.
But then for the bug, it's absolutely necessary that they are there. Does that sound right?
Yeah. I mean, yeah, we can get to this when we kind of move on, but I have
Okay,
some thoughts about that. Yeah, no, but I think you're absolutely right. Yeah.
Okay. And so another experiment that I thought was really interesting is they also did replacement experiments with regular different intestinal bacteria such as E. coli, Bacillus, and Burkholderia.
Yeah, Burkholderia, I think it's I'm trying to remember. It's another symbiont of an insect. So they I think they were trying to pull a not genetically related but lifestyle related microbe to try to see how broadly could you do this swap.
All right.
And those didn't work. So
no, they didn't.
On E. coli and Bacillus and Burkholderia for the stink bugs. The stink bugs do not like.
And the stink bugs ended up with like they all looked, the ones where they did the replacements and they didn't work, they all had the same phenotype as the sterilized ones, where it was just like yellow and crinkly.
Yeah, they don't look happy. Sad stink bugs.
All right. And so if I understand what they did next is, so this was confusing for me. They when they were talking about the environmental symbiont pool.
Yeah.
And then they had these 5 conclusions that they came up with from these from the symbiont pool. So it was hard for me to understand the particular logic for this. So I wonder if you might help me with that, Laura.
Sure. Yeah. Yeah. So so what they'd done was they'd said, all right, we tried to do a replacement with specific strains that we have identified, we know what they are. We know this one's Pantoja from soil. We know this is E. coli, so forth. So then what they wanted to do next was they were like, all right, so this kind of tells us the fact that you can do all the swapping. What you just said, basically, which is this suggests to us that perhaps these stink bugs could potentially, even now, acquire symbionts from the environment, or at the very least, this certainly seems to be like where these lineages originated. And so they then ask the question, which is just like, well, what happens if you just expose some of these eggs to soils from the locations that we sampled? So we're going to assume that there is a population of soil microbes in there. There are probably some of these Pantoja that are related to our symbiont lineages of interest. So what we want to see is, if you just if you don't specifically pick out a strain to inoculate the egg sacs with, if you just have these egg sacs, you incubate them with the soil, and then when the nymphs hatch and the nymphs are around the soil, the thinking is that rather than picking up symbionts from the egg sacs, they're going to pick up microbes from the soil, and then there's going to be some kind of a selection process whereby the symbionts that picked up a suitable lineage that can serve this symbiotic role for them, those are going to do well, and then the rest of them are not. So by doing that, you can kind of start to figure out what lineages that just randomly come from the soil can actually sustain a nymph through to adult emergence, which is a measure of fitness for the relationship.
So they put the larvae in with soil microbes.
Just soil. Just soil. Not even, yeah, just soil samples, yeah.
So then from those soil samples to see if the larvae could acquire those same microbes that were in the soil, and then
yeah, so they basically, so they surface sterilize the eggs, and then they put the eggs with, and then they had the symbiont-free newborn nymphs with soil samples. So rather than picking up symbionts from the egg sacs like they normally do, they're going to pick them up from the soil. And then as we said, you're going to have this selection process, which was actually rather dramatic. So they said they did 1,000 newborns that they subjected to this particular experiment, and 91.5% of them died as nymphs. So 91.5% of them did not, successfully pick up a lineage, a bacterial lineage that could help them. So that tells you that this is probably not the way that these stink bugs are acquiring their symbionts actually in nature. That the vertical transmission is a far more faithful way of transmitting a helpful symbiont lineage than just picking them up from the soil. But 85% of them, 85 or 8.5% of them did reach adulthood. So somehow or another, those successfully picked up some kind of a lineage that was able to serve the symbiotic role that would have otherwise been served by the lineage that had been sterilized away from the egg sac. And then some of those were kind of puny, but 71 or 7.1% were of normal size and green in color. And so they went and took those 71 and they said, okay, well, who have you got? So you 71 that survived, like the less than 10% that survived this experiment, what lineage did you pick up from the soil that's enabling you to emerge as an adult successfully? And so what they found was they actually found that they found lineages C, D, and E at reasonable numbers. And then they found 6 lineages that they just called X, because I don't think they managed to identify those. They just were able to place them into Enterobacteriaceae, and they said they're phylogenetically distinct from the 6 lineages that they'd previously been describing. So these are not found as symbionts in natural populations. They're soil microbes that somehow or another in this experiment were able to actually help the newborn, the nymphs mature to adulthood. Now, granted, this is a fairly protected environment, so I don't really know if you did this in nature, how well that would work, but it's still cool to see how broadly the symbiont role can still be fulfilled for these stink bug populations. That's, I think, the kind of cool outcome of that particular experiment.
So is that more of an adaptation on the whole part, or is that more of an adaptation on the bacterial symbiont part?
I think at this stage you could argue that there is, I would almost argue at this stage that there is no lifestyle adaptation happening quite yet. I think what you might be looking at is I think what you might be capturing here is the idea that the genetic landscape of some of these lineages in soil, these bacterial lineages in soil, they are going to have within their genome the genetic capacity to fulfill a symbiotic role for the stink bugs.
Okay.
So I don't know with this experiment that you would actually argue that there's adaptation in the stink bug side towards having a symbiont, clearly. But getting specific for the symbiont, I don't think that's happened yet. It's probably more that there's a functional capacity that they need. It doesn't really matter what lineage provides that functional capacity as long as that functional capacity is there. But from the bacterial lineage side of things, it really sounds like, you know, if these stink bugs can pick up things from the soil that are able to fulfill these roles, there's not much adaptation going on along the soil microbe side of things quite yet. So yeah, I think you're right that there's adaptation on the stink bug side of things, and I think they're trying to work out like where are we in the road towards being an obligate symbiont with these soil bacteria.
So, I mean, like when you just mentioned that, like, in terms of the functional capacity, what I was thinking is that in terms of the bug, the stink bug, if it's evolutionarily beneficial for it to have this functional capacity to have an adaptation for anything or a wide variety of bacteria that could replace this role. I mean, even if you think about it, like 71 out of 1,000, that's still almost close to 10%. So 1 in 10, that is 1 in 10, right? 1 in 10 of 1 in 10 of every stink bug has the capacity to adapt to another symbiont rather than using the one that's been vertically handed down. And I think that capacity, in a sense, within an evolutionary pool is very useful to have.
Yeah, I see what you mean. Yeah, I agree with you. I think that's very true. I think the 1 in 10 number is probably less that, well, okay. It could be that those are the stink bug individuals that are going to fare best if they have to pull a symbiont from the soil. Or the alternative hypothesis is that that's just stochastic. That it was just random chance. All stink bugs are the same in terms of their potential to acquire a lineage, and then those 10% just happen to be the lucky ones that circumstances aligned that they got one that would work. So in fact, those would be two interesting competing hypotheses to pursue. Is there something going on within the stink bug population that you have a subset that are better able to acquire symbionts from soil, or is it that it was a stochastic process and that's why you got your 10% and so forth? But yeah, I think so what they're trying to get at here is the big question of what's the originating infection event? And you know, at some point it becomes evolutionarily beneficial to, this is why you get this vertical transmission of these symbionts, because if the insect population has evolved to the point that it's dependent upon a symbiont in order to thrive, then it makes sense that evolutionary pressure is going to push this insect population towards having some mechanism for ensuring transmission of the symbiont to the next generation. So in this case, it's the egg sac. So they deposit it on the egg sac, and then there's a behavioral response where the nymphs kind of forage the egg sac to get the symbiont. In the system that I studied, it's the fact that you can find the symbionts in the oocytes. So that guarantees vertical transmission, and there's just never a point that those insects are without those symbionts. And I think that's kind of what they're getting at here is where is the point at which, or is this population going to become committed to one particular lineage? Like what happened in the mainland? I mean, this is a question they didn't really get at, which I thought was interesting. What happened in the mainland population that all they've got is A? Like why is that the case? Like what's going on with A, symbiont lineage A in the mainland? Yeah, I don't know. It's cool.
Well, did they actually say which specific bacterial genotypes were in the mainland compared to the islands? I'm not sure if I
Bacterial or a host?
No, no, a host.
I did not get a chance to actually look at that. I wanted to look at that supplementary data, and I did not have a chance to. So that they may answer that in their supplement. I don't know, but that would be the right thing to look at, which is, what's going on with the host genotype on the mainland.
So we are close to the end, and I'm trying to see if the last experiment with the so with the symbiont replacement. So this one was just, they did symbionts from different types of stink bugs, and then replaced them in with the stink bug. Then again, found that even in different species, symbionts from different species of stink bugs, they were still able to do the job of the original symbiont that they isolated with. Oh, sorry, they put in. No, wait. They put in B, C, D, E in other stink bugs. Is that right?
So they found, so up to this point they'd been working with one stink bug species. And then they said, well, there's other stink bug species, which is like, oh, that's true. And so they went looking for what symbionts those had. And what they found was that they found the phylogenetic tree. I'm just going to do there we go. So in the phylogenetic tree, what they found was that basically they found different members of the lineage in all those different stink bug species. So like here is Symbiont D, the lineage, and they basically said, oh, hey, we actually found, here's our Symbiont D lineage from the stink bug we've been working with. And then here, in fact, is Symbiont D lineages from a bunch of other stink bug species. Same here for Symbiont E, same here for Symbiont C. So what that allowed them to do was to say, well, I kind of wonder if we took Symbiont C, the lineage, from a different stink bug and tried to replace the symbiont in our original species we were working with, would that work? Because basically what they said was we took a stink bug population that was typically had symbiont lineage B, and then we replaced it with C, D, and E from our own stink bug species. But what if we take C, D, and E from a different stink bug species and try to replace it? Then what happens? And basically what they found, what happens is it works pretty much just as well, with the maybe kind of sort of exception of E, which was a little wobbly. But that gets back at the question of there's within host species, you're getting some crossover in terms of lineages. Like what we're not seeing with this tree is that we're not seeing what I'm used to seeing, which is pretty strict co-speciation between host species and symbiont species. You're seeing that still there's a lot of, they use the word promiscuous a couple times. I think that's a good way to describe that. So they're really catching this at a very interesting stage. One of the things that I was a little, so they say their first sentence in the discussion is that they've described an unprecedented snapshot of currently ongoing evolution from environmental free-living bacteria to obligate uncultivable bacterial mutualists. And that I kind of went, yeah, what the hell? I mean, sort of, but not, there's a lot here to start building towards that argument. But I don't know. You could probably conclude pretty safely that the common ancestor of all of these lineages was an environmental free-living bacterium. But it's just tough to catch this happening in real time, kind of a thing. So
they're saying they're kind of in the middle of this symbiotic mutualistic process, and you're saying you can't really say where in the timeline you are. Is that what you mean?
Yeah. Yes, that is what I'm saying. So we don't ultimately know the fate of what's going to happen with these lineages. I think their expectation or their prediction would be that, if you could hop in your little time machine and zoom ahead, probably you'd want to do more than just sample stink bug populations in Japan. But if that's what you decided to do, I think they would predict that you would find the A and B lineages looked quite a bit like Buchnera does now, for instance. Their genomes would have become streamlined. There would be an obligate relationship. You wouldn't find lineages A and B just randomly in the soil anymore, which is true for like Buchnera and Blackmania and these, what we tend to think of as primary symbionts. You don't find them anywhere else. Like, you do not find Blackmania anywhere else but in the carpenter ants midgut. That's where you find them. And other ants too. I always say carpenter ants, and then I get in trouble with ant systematists, so I'm sorry. But here, I think what they're saying is you can find these lineages still making a living or having a lifestyle that's not just in a symbiotic relationship with this insect. And I think their prediction is that that's where at least A and B are headed that way. Maybe not. I don't know. But it's still it's a neat system. So I'm sure they're going to keep working on it. I hope they do, because I think there's a lot you could mine out of this. I would not be surprised at all to find that there's a genomics paper coming down the pipeline from this, because that's the next question, which is what's in these genomes. So,
well, I think that just the genomic factor the species factor in terms of where it almost looks like that these guys, that A and B could diverge off and then become mutualistic. Mm-hmm. And then, but then because of the restriction of an island environment, where the fact that, okay, you're limited, you're not as, you can't be as selective because you've got to get what you can come in contact with. So then being more adaptable on an island is probably more beneficial. So it almost seems like the island populations will remain more free living, while these guys kind of diverge off and become more mutualistic in the future.
Yeah, that's an interesting hypothesis. Yeah, you could really ask a lot of questions about gene flow and about just from a pest control perspective. How much do the stink bug populations, like how much divergence do you see in stink bug populations on the basis of geography? Like, is there a lot of exchange between the mainland and the southwestern islands of stink bugs? Like, do they make it across? Do they get carried in boats? Like, or do you find that you're saying you're getting like Darwin's finches, like you're getting all these little specialized stink bug populations, and then you would therefore expect to get specialized symbiont populations. No, it's a really cool system.
Well, I'm glad you brought up pest control, because that that could even be the next step, which is if you find out the pathways and how these how the stink bug symbiont helps the stink bug metabolically, you can target those metabolic pathways and prevent stink bugs from ever reaching maturity, which also is very cool.
Yeah, and you have just said what was always like one of the opening things that I said whenever I talked about my postdoc work, which was always like, here's why we're interested in this on a practical level. So, yes, yeah, absolutely.
Yes, this is practical.
Yeah, I promise it is.
That's very interesting. Okay, Laura, is there anything else that you wanted to bring up in our?
No, I think this was your suggestion, if I remember correctly, and I really liked it. And technical difficulties aside, I'm really glad we got it up and running, because I really enjoyed reading this paper. It was a nice kind of flashback to what I'd worked on before. It made me nostalgic. I was like, oh my! It's like when you leave a system, then you're probably not ever going to come back to it, and you just kind of have a little moments for like all the questions that were unanswered that bopped around in your head for however many years. You're like, I hope somebody gets to those someday. And so it was nice to read this, because certainly that was something we thought about a lot, which is the evolution of these symbionts and kind of the trajectories that they're on. So I really enjoyed reading it.
It also still sounds like you're still investigating different types of symbiotic relationships or symbioses.
Yeah, with the predatory bacteria for sure. Yeah, I'm trying to pull some of what I learned from my postdoc into that too. But
So it's just it's a broad, you get the broad picture of different types of bacterial relationships. And then I think in that gives you a better context of how bacteria interact with each other. And so that's also why I enjoyed the paper as well, which is the more that the more that I understand about how microbes interact with each other, the more I learn that it's not as simple as we think it is. It's like we can say that, oh, okay, these are obligate mutualists, but then these guys can also survive in soil, and it's even though the stink bug can't survive, but the particular bacteria can still survive in soil. Or we have no idea where in that evolutionary relationship is. Is it early or is it late? And we're just so limited in the number of the tools that we have to be able to decipher that.
Yeah, tools and time too, you know, because these are long real-time experiments. You can't you want to be able to like hop around in time so you can see what's happened. But
And you gotta hope that you have long-term funding so that you can continue to follow each part in that picture, or else you're just gonna lose a giant chunk of the entire story, and that would be terrible.
yeah, absolutely. Absolutely. That's a good point. Yeah.
Okay. Well, thank you for joining us today. Thank you for being patient with our technical
Oh boy,
difficulties.
no kidding.
And so, as always, we'll announce our different papers for the next month. And
I think we're looking for a, not to, I'm sorry to interrupt you.
yeah,
I think we're looking for an April paper. I don't have one on the docket. So if anybody is out there, we've had a couple people contact us about joining the Hangout. Unfortunately, with our technical difficulties, I don't know if that drove anyone away. I really apologize if that's the case. But we're always excited to have people join us, grad students, postdocs, undergrads, you know, people who are in the professoriate, people who are interested scientists who are not in academia at all. Please, you know, if there's something you run across and you think, wow, this is on my to-read list, but I need an excuse to read it and I'd like to talk to people about it afterwards, hi, we are your people. We're taking suggestions for April if you have a paper you'd like to read. I'll turn it back over to you. Sorry to interrupt.
no worries. And like Laura said, we welcome people of any different types of backgrounds, and you can just join us. You don't necessarily have to have read the paper down to the last detail. We won't nail you if that's what happened. But we want to have a great discussion about microbes, and also have this mutual symbioses of knowledge with each other, so that we can learn and understand more. And and that's it for today. And so thank you for joining us. We will see you next month. Bye.